A cytoskeletal protein might be involved in promoting breast cancer by making cancer cells acquire some features of stem cells. Researchers have found that FASN, a protein that bundles together the actin filaments that serve as the backbone of cells, is important for keeping breast cancer stem cells alive. The results shed light on what is going on inside cells as they break from normalcy to become cancerous, and highlight FASN as a potential diagnostic marker and therapeutic target. One reason why cancer can be so hard to treat is that certain tumor cells are resistant to chemotherapy and can self-renew, providing an endless supply of cells to reseed tumors. They're known as cancer stem cells because they hijack several properties of stem cells, like expressing transcription factors that allow for self-renewal, to push their cancerous agenda. FASN is found in higher amounts in tumors and has been linked to breast cancer chemoresistance and spread, two key features of cancer stem cells, raising the possibility that this protein might promote cancer by regulating cancer stem cells. To find out if that's the case, researchers first experimentally lowered the amount of FASN in a human breast cancer cell line. When treated with chemotherapy, they found that these cells died more readily than those with the protein. The surviving cells, which were more common among cells with FASN, were more likely to be cancer stem cells, expressing high levels of certain markers. The reverse experiment, giving cells extra FASN, allowed more cells to survive chemotherapy and increased the number of apparent stem cells. When the scientists checked for stem cell-related transcription factors, they saw a similar trend. The cells without FASN had reduced amounts of self-renewal factors, while cells with extra FASN had higher amounts. The researchers then looked to see if FASN was associated with cancer cell invasion and spread. As cancer cells prepare to spread, they become more spindle-shaped and mobile. In a cell model of this transition, scientists found that FASN levels increased, while removing FASN reversed this process. FASN was also found to be critical for tumor formation in the petri dish, strongly supporting FASN's role in regulating cancer stem cells. But the real test is whether FASN can help cells self-renew and create tumors in animals. To find out, researchers injected small doses of breast cancer cells into mice with compromised immune systems. Just 50 cells with FASN were needed to produce tumors in a third of the mice. Without FASN, though, none of the mice developed tumors with so few cells. How might FASN be doing all this? Notch is an important signaling pathway that controls cell fate in the breast. The scientists found that in cells without FASN, Notch signaling was low, and that signaling was necessary for robust tumor formation in the petri dish. In fact, blocking Notch signaling made cells with FASN behave as if they didn't have the protein. They were less able to form tumors and more likely to die after chemotherapy. The findings suggest that FASN makes it easier for cancer stem cells to form tumors and stay alive by activating the Notch self-renewal pathway. Paying attention to FASN levels in patients might be a valuable way of monitoring cancer and personalizing therapy. And one day, targeting FASN with a drug, in combination with other chemo, might even provide a more effective way to treat the disease.